afternoon, Dr. Naomi Visanji. Doctor, you can come up, that's fine. Can I say nice things about you before you start? All right. Dr. Visanji received her honors BSc in neuroscience at the University of Nottingham in the UK in the Department of Biomedical Science. She completed her PhD in pharmacology at the University of London. From 2004 to 2007, she was a postdoctoral fellow at Toronto Western Research Institute. And she is currently training under the supervision of Dr. Lily Naz-Mizrahi at the University of Toronto. Naomi has always been supportive of Parkinson's Society Canada's work and is willing to help and participate in research-related events. We're thankful for her dedication. Her strong commitment to people with Parkinson's was exemplified this past September when she put together a super walk team and sent out a challenge to all current and previously funded Parkinson's Society Canada researchers to encourage their labs to do the same. She posted the following message on Facebook. Dear Superwalk participant, I'd like to personally thank you for taking part in Parkinson's Superwalk 2010. Your commitment and the funds you raise provide vital support services, advocacy, and education to living well with Parkinson's every day. This year, I decided to form a team with my University of Toronto colleagues and participate in the Toronto Superwalk. It was a moving experience to witness the hope in action, walking alongside people with Parkinson's, supported by their loved ones. Thank you for your stellar fundraising efforts and enthusiasm. Participating in Parkinson's Superwalk as a neuroscientist is very specializing in Parkinson's. I hope that one day we will find a cure. In the meantime, we will continue to focus on learning as much as possible so that we can intervene earlier and to improve the quality of people's lives. I know we are in for a treat today because two of my colleagues attended one of Dr. Visanji's presentations that she did for us out in Cobourg, Ontario. So she also travels for us. And their feedback, as well as the other anecdotal reports we received, were along the lines of something like this. For the first time, I really understood what Parkinson's is all about. So please give a warm welcome to Dr. Naomi Visanji.
Good afternoon, everybody. I just want to say it's an absolute pleasure to be asked to come here today and speak with all of you. I can say that our group that participated in the Superwalk in September, we really enjoyed the day. And it's quite unusual for us to actually be able to meet people with Parkinson's, meet their caregivers, basic scientists who are kind of locked up in the lab, and we don't meet patients. So it was a really good experience for all of us. We had 3 or 4 of the younger students who have just sort of started came along and definitely got them interested in studying Parkinson's. So yeah, it was very inspiring for us. And thank you very much. Okay, so as Sandy said, I'm actually from England, you can probably tell. And I've been here for nearly 7 years now. I moved over here to do my postdoctoral research with Dr. Jonathan Brocci, and now I'm at the University of Toronto. And I was lucky enough to receive a 2-year basic research fellowship from the Parkinson Society a couple of years ago. I just wanted to let you know that I know that a fair amount of the money that you raise from things like Superwalk, they go to this funding of basic science, and it's a really fantastic program. Something that I hope the society continues supporting young scientists such as myself, and giving us the ability to do the research that we really want to do through having our own funding. So I'm going to start by giving you a little bit of background to Parkinson's disease. It was actually first characterized in England in 1817 by this guy James Parkinson, and he wrote what he called an in the department where I did my PhD. And James Parkinson described Parkinson's disease as an involuntary tremulous motion with lessened muscular power, in parts not in action, and even when supported, with a propensity to bend the trunk forward, to pass from a walking to a running pace, and with the senses of the intellect being unimpaired. And I'm sure that's a description that many of you can recognise. It's now been updated slightly, and so the current definition of Parkinson's is given below. So this slide here, I wanted to try and explain what's actually going on in the brain with normal movement generation, so that I can then describe what happens when this system becomes impaired. So what you see here is a brain, and sorry, it's a bit of an awkward angle. So what you can see here is the brain, and then on the right-hand side you see some boxes that basically represent different nuclei, clusters of areas in the brain that are involved in movement generation. And what happens when you actually think about wanting to move? Before you move, you think about it, and all of that's going on in your cortex. That's your real thinking area. So you have this excitatory input from the cortex, which is the top green box, and that projects to an area called the striatum. And once it reaches the striatum, the signals are then integrated and then sent to this area called the basal ganglia, which is a set of other nuclei in the brain that allow you to actually move your muscles. Now this area called the striatum is kind of a junction box. So you've got these inputs from the cortex saying, I want to move, and you're thinking about moving. And at the same time, you've got some cells from the substantia nigra that project from the substantia nigra to the striatum, and they actually secrete this neurotransmitter dopamine onto the cells in the striatum, the junction box. And that allows them to integrate what they're doing and to coordinate your wanting to move with the actual messages that go out to your muscles and allow you to move. And what happens in Parkinson's disease is that these brain cells in the substantia nigra that produce and secrete the dopamine into the junction box, they actually start to die. So on this slide what you see is a normal brain on the left, and you can see those dark patches. That's actually the cells in the substantia nigra. They actually produce dark pigments, so they're really easy to see. And in the smaller little box inlay, you can actually see that's just a high power magnification of those cells. And that's the substantia nigra where these cell bodies are, and then they project to the striatum, which is shown below. That's actually a PET scan. And where you see that red signal, that's the striatum, and that's showing us that red signal is a signal of the dopamine that's being released there. And then you can see on the right-hand side, in the Parkinson's brain, up in the top in the substantia nigra, you've lost a lot of those dark cells. They've actually died. And as a result, the region they project to in the striatum, you can see we've lost that lovely red signal of the dopamine. So dopamine is no longer being secreted in this junction box, and it basically creates a disconnect between your desire to move and your ability to move. So that's what's happening. So long time ago, people started to wonder why these cells die. And in 1912, there was a German neurologist, his name was Louis. He actually discovered that there were these strange clumps, these strange Lewy bodies, they're called, in the cells in the substantia nigra of people with Parkinson's disease. Interestingly, I just wanted to point out that Louis is the guy in the white coat on the right, and then in the centre of the picture is another person, Dr. Alzheimer. So they were actually colleagues. It's pretty amazing that they work together. So anyways, Louis discovered these Lewy bodies, and now basically everything we do with Parkinson's research centres on what are these Lewy bodies. what causes them. So in 1919 it was shown that they were present in the substantia nigra, and since then we've been studying it. And finally, in 1997, scientists in the States discovered what is inside the Lewy body. And they actually found that the Lewy body contains very, very high amounts of this protein called alpha-synuclein. And that's basically what this image you see on the right, that brown circle, that's the Lewy body, and that has been stained for alpha-synuclein. So the brown signal that you're seeing is alpha-synuclein. And synuclein is present in all neurons. It's a much needed protein, obviously. We don't know what its function is, but it doesn't normally aggregate like this. So this is, the kind of underlying pathology of what's killing the cells, basically. So, as I said, they've been shown to be very numerous in the substantia nigra. But what's happened more recently is some research has grown looking at the spread of Lewy bodies through the brain. And they've actually found that they're not just in the nigra. So another German scientist, a guy called Michael, he's actually put together a new paradigm on the development of Parkinson's disease. And what he did was he basically looked at the progressive development of Lewy bodies throughout different areas of the brain. And they've shown that the disease actually spreads from one area to another. So they put together this model that's now called Braak staging. And it's basically based on where you see those Lewy bodies. And in stage 1 and stage 2, this is sort of preclinical stages. And the Lewy bodies are actually found down deep in the brain stem, in the medulla and the olfactory bulb. And stage 2, you start to see some more Lewy bodies in these areas. And they call this stage preclinical, but actually this is when people tend to see symptoms in retrospect. So once they're diagnosed with Parkinson's, and you say, well, did you notice anything happening with your sense of smell a few years ago? And people say, yes, yeah, I did. So that's actually stage 2. It is clinical, but most people don't realize it at the time. But actually, because you've got these Lewy bodies in your olfactory bulb, and that's the area of your brain that controls your smell, it seems that they're impairing your ability to smell. Maybe several years before you actually go on to develop some of the motor complications of Parkinson's. So then the Lewy bodies spread a little further, and we start to see them in the substantia nigra in stage 3 and stage 4. By stage 4, you have quite a lot of Lewy bodies in the nigra, and this is when those motor symptoms start to manifest. And then finally, the Lewy bodies start to spread to other areas of the brain, the amygdala, which is an area of the brain which controls emotional and motivational aspects, and finally, they can reach the cortex and start to have patients and suffer from dementia and other problems associated with the cortex. So this is a relatively new concept, and when it first was proposed, some people didn't really like it. But really now it's become very well accepted. One of the main reasons for that is from some new evidence that came out probably 2 or 3 years ago. So basically what happened was, a couple of years ago, one of the first patients to receive the stem cell grafts, the fetal cell grafts into the brain, one of the first patients to receive those actually passed away, and scientists were able to look at the brain and see what was going on there. So this patient, she'd had Parkinson's for 22 years when she underwent the surgery. She had her surgery in 1993, and at that point she did have very, very severe motor fluctuations, very severe dysphagia, and she was unresponsive to medication. So they transplanted fetal dopamine cells into the striatum, so right into that junction box that we were talking about. And by 1997, 4 years after her transplant, she had much improved. Her UPDRS scores were down, her dyskinesia was a lot milder. But unfortunately, this benefit was pretty short-lived. By 2004, her condition began to worsen, and she passed away in 2007, 14 years after she had the surgery. So So this is actually tissue taken from her brain. And what you're looking at is on the far left-hand side, you're looking at the area where the graft actually was given. And TH and VMAT2, they're basically just two proteins that are markers for dopamine cells. And you don't normally have dopamine cells in that junction box. So every brown cell that you see there is one of the fetal cells that's been grafted in and is surviving and living, and because it's expressing these two proteins, we know that it's healthy. So this was good news. They were, the scientists were quite excited that the cells lasted for 14 years. And then on the right, what you see, the panel on the right, the two sections that are on the left of the panel on the right. Sorry, I can't use the laser pointer. What you're seeing there is Lewy bodies in the substantia nigra. So this is the area where the cell bodies are, where you see those dark signals. And that's stained for Lewy bodies, the alpha-synuclein. And that would be normal. We would expect to see Lewy bodies in the nigra of somebody that had Parkinson's. But on the very far right, what you're looking at there is the striatum. So again, back to the junction box, where the healthy fetal tissue was put. We actually see Lewy bodies there. And this stuns the scientists because these cells are only 14 years old. They come from, it's a fetal graft. You would never expect to see Lewy bodies, a sign of Parkinson's disease, in cells that are only 14 years old. So what's happening is somehow this toxic signal is actually infiltrating from the patient's diseased tissue. It's infiltrating this healthy tissue that's put in there. And you're seeing a spread of Lewy body pathology. And this was a really big surprise to the scientists, but really gave credence to the Braak staging, basically proving that Lewy bodies can spread from one cell to another, from an unhealthy cell to a healthy cell, and equal to just a 14-year-old cell. So it's really convinced the field that the Braak staging is real. And a lot of research is going into this now. What is this toxic factor that's spreading the disease from a healthy cell? From an unhealthy cell to a healthy cell. And at the moment, people feel that the most likely candidate is this protein alpha-synuclein that we talked about. And they've shown that it can be released from cells, it can be taken up by neighbouring cells. And there's a lot going on in that field at the moment. So, I wanted to sort of say that Parkinson's disease is a disease, in many ways, it's got a real window of opportunity for some sort of therapeutic intervention. And there's two reasons for this. Basically, by the time the motor symptoms of Parkinson's disease present, when you've got that stage 4 Braak pathology, already 50 to 60% of those dopamine cells in the substantia nigra have died. And this tells us that the brain has an absolute huge capacity to compensate. You can lose more than half of the cells in the substantia nigra without having any symptoms at all. So the brain is doing a really good job of trying to make up for that loss. So lots of people are looking into ways to help the brain do those things that it's trying to do. And the other thing is that the Braak staging tells us that by the time your symptoms appear, the disease has been there for who knows how long, but at least 7 years, probably a lot longer. So if we can find a treatment and we can give it to people before their symptoms present or when they very first do, we can try and stop the disease developing any further. And there's not many diseases that have this window of opportunity. So I think there's a lot of hope out there in terms of being able to do something effective there. So what's happened really since this has all come about, since the Braak staging, there's been a real shift in research in Parkinson's disease, and people have stopped just looking for other dopamine agonists, other symptomatic treatments, and they're really shifting to looking into preventative therapies, neurotropic factors, something to prevent the cells from dying, so that you wouldn't even have to have symptoms in the first place, basically. So I'm going to try and give you basically a kind of whistle-stop tour of some of the things that are going on looking at neuroprotective therapies. I'm going to talk to you about the need for biomarkers, because obviously if we're going to come up with a neuroprotective treatment, we need to identify the people that need it before they get the symptoms. I'll talk to you a bit about some work on neuroprotective therapy that I worked on, and also on some novel antidyskinetic research that's going on. So I'll start with biomarkers. What I've put up there is the NIH definition of biomarker. Basically, a biomarker, it can be almost anything. It can be specific cells, it can be molecules, a gene product, a hormone, anything that gives a clue as to what might be going on in the brain. An example of a classic biomarker is cholesterol. We all use cholesterol now as a biomarker for what's going on in our cardiovascular system. So biomarkers, basically, there's three main characteristics that they need. They have to be easy to sample, so blood, urine or saliva would be ideal. The speed of testing is important. Nobody wants to have to wait months and months for their results, so the quicker the better. And also, obviously, it has to give a really accurate measure of what's going on in the brain. And it's difficult to satisfy all of these three criteria, obviously. And at the moment in the Parkinson's field, there's sort of three avenues of research for biomarkers. The first one is brain imaging. So an ideal Parkinson's biomarker would really give us a measure of what's going on, how much dopamine do you have in your striatum, or how many of those cells do you still have in the micro. Currently, this can't be measured directly, and it can't be measured non-invasively. There is a technique out there called the DAT scan. Now, that's actually being used for some Parkinson's diagnosis in Europe. And basically what it does is you would receive an injection of a radioactive compound, so it's fairly invasive. And this radioactive compound then it labels the areas in the striatum where dopamine is released. So that is a dopamine transporter, and it's a protein that sits in the striatum on the cells that are releasing dopamine. And you can see here the images, you can see that sort of red signal that you're probably familiar with by now, giving us levels of, sort of putative levels of dopamine in that area. The problem at the moment with this is that it's pretty expensive, it's not completely accurate, and it doesn't really, it's still an indirect measure. We're not measuring dopamine, we're measuring something that's associated with cells that secrete dopamine. So it's still pretty, it's not perfect basically, which is why it's not available everywhere. A second avenue of biomarker research at the moment is indirect biomarkers. So this would be that something that we don't know what yet in the blood or the urine or the CSF that may give us a sort of signature of what's going on in the brain. And leading markers in this area are mostly proteins that have been linked to Parkinson's through genetic studies, and one of them being alpha-synuclein that we talked about. And then the final type of biomarker that they're trying to get together is clinical measures. So these would be really, really sensitive clinical tests that would pick up on early disease symptoms. And top of the list there is the olfactory deficits, that's loss of smell that occurs really, really early on. And one that I'm going to talk to you about that's relatively new is actually sleep disorders that might be giving us a clue to Parkinson's disease. So the sleep disorder I'm going to talk to you about is called REM sleep behavior disorder, or RBD, and it's unknown cause at the moment. And it's a really strange sleep disorder. Basically what happens is the dreamer really, really acts out their dream. So when they go into REM sleep, normally when you go into REM sleep, your muscles relax, even though your brain becomes active and you start having your dreams. Your muscle activity is inhibited so that you don't move around. And what happens with these people is that switch somehow is lost. So that when they go into REM sleep, they can actually move around. And when they're woken up, they can usually recall the dream they were having. Often the actions they were performing match what they were dreaming about. And it can be quite severe. We're not talking about just those little jerks that we all have as we fall asleep. I'm going to show you a video now, and that will show you sort of exactly what we're talking about. So these are videos of patients that have gone to a sleep clinic, and they've got an EEG hooked up to them. So as this guy moves around, he sees the wires attached to him. And basically, the EEG tells us when the person goes into REM sleep because the brain waves change. And he goes into REM, and he's just moving around, which is obviously quite unusual, and really flailing about. This guy actually, when he was woken up, said that he was playing football in his dream. Soccer to you guys. He's kicking a ball, he catches the ball and kicks it. You can really see that he's acting out exactly what he's dreaming. And this last guy, when they were woken up, said that he was swatting insects that were in the lab. You can see him swatting them. And I'm sure I don't need to point out to you the dangers of sharing bed with one of these people, but there have been cases where people have been quite badly injured by their partners in this state. So why am I telling you about this strange disorder? Basically what's happened is RBD first became described as a disorder in 1993, so not that long ago. And in 1993 what they found was that 38% of people with RBD developed Parkinson's disease. And as time goes on, as there's more time since 1993 and we've been following these people with RBD, our risk is going up and up. So in 2003, 65% of people who had RBD had gone on to develop Parkinson's. And many groups are reporting this, and I've been told that the incidence now is right up to about 80%. So as these people with RBD get older, there's an 80%, maybe even higher chance of them going on to get Parkinson's disease. And this is a really, really strong link, obviously. And interestingly, what's going on in RBD, it's a relatively new field, but what they do know is that the brain area that's involved in allowing your muscles to relax when you go into REM sleep, preventing you moving around, is the exact same brain area where we see the Lewy bodies in stage 2 of Prague staging. So again, it's another clue that things are going on deep down in the brain and disturbing maybe other parts, and then spreading through there. And we're really excited because we've recruited a postdoc who specialises in RBD to come and work in our lab starting in January. She's going to be looking at this link, so watch this space. So the next thing I'm going to talk to you about is neuroprotection. So as I said, this is becoming a really big field. And I'm sure a lot of you have heard about those clinical trials using the GDNF and BDNF for Parkinson's disease. So I'm going to talk to you a little bit about that. So basically, GDNF and BDNF, they're proteins. They're actually made in the brain, and they help neurons to survive. So neurons, they carry out a lot of complex functions, and they need all sorts of support to be maintained. And GDNF and BDNF play a big role in that. And what you're seeing here is another one of those sort of dopamine scans. And this is a patient before and after they received their GDNF infusion. So on the left, you can see that where the little white arrow is, that's the actual site where the GDNF was infused. And you can see that on the left, there's no red signal there at all. There's not much dopamine signal whatsoever. And then on the right, what you're seeing is the same patient that 24 months after they've had this growth factor infused into their brain, and lo and behold, you've got a nice bright red spot. So dopamine is being secreted there once again. So one of the problems with this is that it's obviously very invasive. Having to have brain surgery, it's far, far from ideal. And this is one of the problems. Both BDNF and GDNF are really, really huge molecules, and they can't cross your blood-brain barrier. So if you had an injection of one IV, it can't get from the blood vessels in your brain into your actual brain tissue because of this blood-brain barrier. Another problem with them is they're proteins, and if you eat a protein, if you took BDNF or GDNF as a pill, your stomach's just going to digest it. So you can't take it orally. So what scientists are trying to do are find ways of upregulating the brain's own natural production of BDNF and GDNF, rather than relying on us synthesizing some and putting it in there, when we find a way of augmenting the brain producing them. And I was really, really lucky a few years ago to be able to work on a small molecule, BYM, also known as cocaine, and look at some studies of that in regards to Parkinson's disease. So cocaine is actually, although it's patented now, it's an extract of a plant. And this plant forms a component of this Chinese medicine that apparently has been used for over 100 years as a tonic for aging. So for 100 years, the Chinese people have already known exactly what they're doing, we're just catching up. And this product has been isolated from this tonic, and it's been shown to reduce age-related declines in BDNF, and it's also been shown to be helpful preventing cognitive decline in Alzheimer's disease. So we along with the company that produced cocaine, decided to look at how cocaine might help the dopamine neurons in Parkinson's disease, and see what it was doing for BDNF and GDNF in that aspect. So these are some data from a paper we published a couple of years ago. And what you're seeing here is some cells in culture. These cells, they're basically dopamine cells. They're taken from rat brains, and they're made into, we grow them in a culture dish, and then we're able to take a look at what's happening with them. And what we do is we use a toxin, which is called MVP+. And if we add this toxin to the cells in culture, they die. So the white bar shows you some just normal cells, untreated with anything, which is letting them grow healthily. The next bar across is where we've added this toxin, and you can see we've killed about 50% of the cells. And to the right of that, you can see the effects of cocaine, and then also BDNF and GDNF on this toxin-induced cell death. And in this paradigm, we treated the cells with the cocaine for 24 hours before we added the toxin. And you can see that we've completely prevented the cell death that's induced by this toxin. And then on the right, what you can see is the actual cells themselves. You can see that the vehicle cell, that's the one for panel B, it's a bright, healthy cell. You can see it's got long neuritic processes. That's how the cells communicate with each other. You can see in panel C that after this toxin treatment, the neurites have disappeared and the cell is a bit duller. And then beneath it, you can see those that have cocaine or those that have BDNF and GDNF. They look kind of like the one that didn't have the toxin. This is a really, really encouraging finding. And what we then went on to do was to see, well, what if we don't pre-treat the cells? Because right now we don't have that option in Parkinson's disease. We normally wouldn't treat someone until they've already got symptoms. So what if we treat the cells with the toxin, wait 24 hours, and then add the cocaine? And that's what you see here. And you can see that the cocaine, even when we give it after the cell death, the injuries occurred to the cells, we were able to bring the cells back, which was a really, really exciting finding. So because it was working so well in our cell culture model, we were able to get permission to do some studies in animals. So what you're looking at here is some studies we did in MPTP-treated mice. MPTP is it's a chemical that was discovered back in the 70s a group of very young people in California all got Parkinson's disease out of the blue. And they all were showing up at the doctors, and they had developed Parkinson's disease in a very, very short space of time, all showing up at the doctors. And what happened was they found out that all of those people, they were actually heroin addicts. And there had been a contaminant in a batch of heroin in California, and it contained this nasty substance, PDTV, and it had killed their dopamine cells in their nigra rapidly. It was really awful, but something that came from it was this animal model. Scientists in California, it's actually a place called the Parkinson's Institute, sprung up as a result of this, and they used this chemical to create animals with Parkinson's. So we can give this chemical to the animal, and it will develop Parkinson's disease very quickly in up to 5 days. It's very, very fast. So what you're looking at here is the substantia nigra over mouse. And so we treated the mice with the MPTP, and you can see the middle black bar. We've killed approximately 50% of the dopamine neurons. So that's the count of the dopamine neurons. And you can see the top picture, panel B, that's a healthy nigra. Panel C, you can see that there's less of those dark brown cells. And then the third bar along is the cogaine. So we treated the animals with the cogaine, and we were able to restore a significant number of those dopamine neurons that were lost. And in this case, we treated the mice for 60 days with cogaine. And what you see here is the striatum, so the junction box again. And you can see that the healthy animals have that. Sorry, it's confusing here, but my signal's green for healthy and red for unhealthy. So the green signal is looking at the DAT, looking at that monitor dopamine release in the striatum. And then you can see that the animals that had the MPTP, that's lost probably about 60-70%, but the animals that had the 60-day treatment with the cogaine, they're right back up there, and their dopamine levels are pretty normal. Okay. And the next thing we did was take a look at the BDNF and GDNF in the brain. And what we saw was that we had this massive increase in BDNF and GDNF after cogaine treatment. So cogaine is taken orally, it crosses the blood-brain barrier, it gets right into the brain tissue, and it helps the brain produce its own BDNF and GDNF. And I called Dr. Birchie yesterday to find out what's happening with cogaine, and he told me that the phase 1 trial in healthy people has been completed, and they're doing a 6-month phase 2 trial at the moment, and they're expecting some results from that in 2012. So we'll know in 18 months or so, I guess, whether that's working out in Parkinson's patients. So moving swiftly on. Longer than I thought. The last thing I'm going to talk to you about is antidyskinetics. So I'm sure many of you suffer with dyskinesia and find that there's not much out there that effectively alleviates that. And I'm going to talk to you about a sort of case study of some work that was done in the UK. So this guy, Tim Lawrence, he was a film stuntman, very, very active guy, and unfortunately developed Parkinson's disease at age 34. And he got really, really severe dyskinesia very, very quickly. And Tim, he was actually a recreational drug user. He used to take ecstasy every now and then. And what he found was, when he took his ecstasy, his dyskinesia went away, even though he was on his peak dose of L-DOPA. So he went and told his doctor this, and some research, interesting research, followed after that. So what you see here, this is a video clip of Tim in his kitchen, and he's just explaining how difficult it is for him to do something that all English people like to do. He's trying to make a cup of tea, and he can't make it because he tends to spill the water all over himself. His dyskinesia is really quite severe and very, very frustrating for him. And then what you're going to see is a second clip where he was in a gym and he's taken his L-DOPA, but he's also had ecstasy. And he's back to doing his stunts. He can do acrobatics that I can't even dream about doing. And this guy has Parkinson's disease and dyskinesia. Absolutely amazing what he was able to do. You can see his movement's not perfect, but it's a heck of a lot better than it was without the ecstasy. Quite inspiring. So the scientists were really, really excited by this, just because it's easily the biggest antidyskinetic effect that had ever been seen. So Tim actually had to go into a lab and undergo some experiments to prove that this was a placebo effect. We all know how powerful a placebo can be. So he went in one day and was given a pill. Nobody knew whether it was ecstasy or not. And on the second day he went in and was given a pill. Again, nobody knew whether it was the placebo or the ecstasy. But it proved that his movement was only smoother when he had the ecstasy. There was no placebo effect at all. So what happened, he had lots of brain scans. People wondered whether the ecstasy was helping with the dopamine somehow. And it wasn't at all. It was actually affecting a different neurotransmitter in the brain called serotonin. And so what's going on now with this is, obviously I'm not suggesting that any of you go out there and buy ecstasy. It's illegal. It's also, it's pretty harmful, especially for people that already have some sort of degenerative process going on. It kills brain cells. It's not something you should take. But scientists, other scientists like the one in the picture in the middle here, they're trying to find a way to manipulate the ecstasy into a way where it's not harmful, but it still has antidyskinetic properties. So, again, watch this space. I spoke to some friends that work on this, and they're hoping to publish something soon. 3 minutes? 3 minutes. Okay, great. I'm going to rush right through. Unfortunately, I can't talk to you about my work, but I was going to talk to you about the work that was sponsored by the Parkinson Society, but what you need to know is it was very interesting. I'll work right to the end and just acknowledge the people that I work with. So I'm currently in Dr. Lilian Hansarati's lab. I'm actually carrying on the work that I was doing because we were very interested in it. I also want to thank Dr. Barucci. I did all of the cocaine work in his lab. And the lady at the bottom, Dr. Collingwood, is someone in the UK that we currently collaborate with on our work that's sponsored through you guys. And also to thank the other people that have sponsored the work I do. And it's the last slide I was going to put up. There's just some things, I don't know if you guys have heard about these different places that you can find out about current research in Parkinson's. But they're really good websites that you might want to take note of. I'll go back to that one. I put my email address on there, and if anybody wants to email me, feel free. I'm happy to answer any questions that you might have. Thank you.